Good evening and welcome to our new seminar series. Thank you so much for joining us here this evening, both here in the McCarthy Auditorium and at home via live stream. Before we start this first presentation, I would like to say a little bit about the purpose of this seminar series and how you can participate in this series. The presentations in the series are aimed to inform our society about important topic for our society and for the well-being of our society. Our increasingly fast-paced world is changing rapidly, and I'm sure I'm not alone with questions that this change brings with it. My personal knowledge across many of these disciplines is very fragmentary. What are the benefits and cons of vaccination? How do I help my child to learn effectively in school? How do current political events affect world economy and stability? Why are our pollinators on the decline? And what can I do to change this? How do drugs affect the human body? And how can I explain drug-induced behavioral changes? I'm sure I'm not alone with these questions, and neither are you. And you can participate in this by relaying questions to us that you have and you would like to have addressed in one of those seminar series. We would be grateful for your input here. The goal of the seminar series is to have experts across different fields address our questions and help us gain a better understanding of those topics, utilize the expert knowledge we have to benefit all of us. My name is Ruth Fabian Fine. I'm the current director of the neuroscience program here. I'm a professor in the biology department, and I would like to introduce my colleague Adam Weaver. We conduct research together and we teach together, and Adam will take the lead on this presentation. Thank you.
Thank you. So, as this is the inaugural seminar of this series, we wanted to, of course, invite you to the next two talks that we have scheduled for the fall semester, both on October 20th with our physics professor Alain Brizard, who's going to speak on aspects of fusion and how that's becoming more and more a likelihood in our near future. And then, of course, on Thursday, November 3rd at 5:30 p.m., in the same room for both talks will be Ari Kirschenbaum, who's going to speak on the aspects of marijuana and its recreational use and how it affects motor skills. So with that, I just want to remind the St. Mike students in the audience that you can scan the QR code to actually enter into a raffle. This is an uplift event and continues in that strong series of events. The overview, we have a lot to potentially present in a short amount of time, but I wanted to just kind of briefly speak to that. First, we're going to talk about the history and prevalence of neurodegenerative disease and disorders, the different types of those diseases, current hypotheses on why neurons degenerate, and because the best understood of those diseases happens to be Alzheimer's, we're going to give a brief overview of that disorder. And then we'll talk about loss of memory in the absence of neurodegeneration. That is, many of us will become forgetful, and that's a normal part of aging, but doesn't necessarily mean that you are destined for a neurodegenerative disease. We'll move on to an introduction to neurons and their important structures, alcohol and its effects on neurodegeneration, limitations of current model systems, and some novel findings that our group, students and professors alike, have found with this invertebrate model system. And then finally, we'll talk about sleep and brain health. So one of the best understood neurodegenerative disorders is Alzheimer's disease. And it was discovered at the turn of the 20th century when a patient, a female patient, Auguste Ditter, who was around 50 years old, was admitted to the Frankfurt Psychiatric Hospital. She expressed an array of symptoms that made it difficult for her to live at home, including sleep disorders, memory loss, and severe mood swings, including aggression, and progressive confusion. And despite the best efforts and the care that she received in that facility, she progressively declined during those 5 years. And a researcher and physician, Dr. Alois Alzheimer, studied her during that time period and documented her progressive decline and the disorder she was suffering from. I do want to point out that she was only 50, so this is what we would typically classify as an early onset form of this disorder, and of course we now call it, after the physician, the Alzheimer's disease. And when we compare the brains of an individual who may have passed away from natural causes, so-called, we have the classic brain shape that we see here, the larger brain volume. But when we compare that with an Alzheimer's disease patient, especially a severe one, as you see to the right, there's significant loss of brain tissue. And in some examples, some samples, the brains actually become darker over time as part of that neuron loss. As I'm sure we're all aware, there's a lot of challenges associated with neurodegenerative disease, but I want to summarize a few. The first, and probably the most important, is that neurodegenerative diseases are one of the top 5 causes of death around the world, let alone in the US. And it's exceedingly difficult for patients and caregivers in terms of time, the time of spending time and care that's needed for those patients, the costs, both financial and emotional, toil, the effort, as well as the loss of function over time. And another challenge with neurodegenerative diseases is that they're often very difficult to diagnose. The early stages are really hard to differentiate from normal aging processes. And as I will discuss later, we're actually now understanding that many of neurodegenerative diseases are an increased amplitude and speed of onset of normal aging processes in some ways. But once the symptoms are clear, the diagnosis is in hand, often the patients have progressed so much so that it's difficult to actually treat. And so they end up typically treating symptoms and trying to reduce them wherever possible. And then finally, another important challenge is that 2 different patients with a particular disease, Parkinson's or Alzheimer's, may have slightly different underlying causes due to genetic risk factors, environmental factors, and so forth. So when you have what we would call a syndrome, a collection of symptoms with potentially different underlying causes, it makes it challenging to actually treat and fully understand. When we think about the role of aging, aging is the primary risk factor for the onset of neurodegenerative diseases. When we look at, when multiple studies have looked at the age in which dementia comes upon a patient and the percentage of patients of those age groups that express the disorder, we have a graph here with multiple lines from multiple different journal sources. And as we see across the bottom here, as we increase in age, starting at 65, going up towards our 90s and 100, the prevalence of dementia greatly increases. And by the time one reaches their 90s, upwards of 40% of the population will express some form of dementia. When we look at other disorders like Parkinson's, it's a little bit more complex and maybe not as clear. Some journals have argued that with increasing age, there's an increase in Parkinson's prevalence, but not all have found that to be the case. So it's a little more complex. So we know that the rates of diagnosis of neurodegenerative diseases are increasing over time, and the primary reason for that, of course, is that we as a population around the world are living longer. And so, 100 years ago or more, one of the underlying causes or typical causes of death might be tuberculosis. Now we don't typically die from tuberculosis, we live longer, and with a longer period of time, we're able to experience new disorders and new diseases, and these neurodegenerative diseases are some of those that we experience. So I want to point out, though, by 2030, it's estimated that 1 in 5 Americans will be over the age of 65, so we're expecting increasing incidences of neurodegenerative diseases as we move forward, as our population ages. When we think about major types of neurodegenerative diseases, there's common forms, that is, have higher prevalence rates, and then there's the others that are no less important, but just unfortunately are less studied. Some common ones are Alzheimer's disease, that in the US we estimate that upwards of 5 million within their population have some form, mild to severe forms of Alzheimer's. And as you might know, it has significant learning and memory components in terms of deficits. When we look at Parkinson's disease, there's a lower incidence, around 1 million, and some of the major hallmarks of that disease are tremors and loss of motor control. Multiple sclerosis is about 400,000. It's an immune-associated disease that causes what we will call demyelination, that will just be described later. But it leads to early symptoms like numbness from the body, and then eventually later issues with motor function. When we look at another disease, amyotrophic lateral sclerosis, abbreviated ALS, you might know it as Lou Gehrig's disease. That's one that causes progressive loss of neurons in the motor pathway, that activate muscles, and causes motor loss and paralysis. And Huntington's disease also affects motor coordination, but also impacts cognition as well. And they both have around 30,000 in the US that suffer from those disorders. Looking at the other disorders, other forms of dementia, that is, besides Alzheimer's, include dementia with Lewy bodies, alcohol-induced dementia, and frontotemporal dementia, and those typically lead to severe confusion and hallucinations in many cases. And when you combine those forms of dementia with Alzheimer's, they estimate between 1% to 2% of any given population will actually have that. When you get above the age of 65, the incidence rates go up dramatically. You have other disorders like Guillain-Barré, spinal muscular atrophy, and motor neuron disease, all of which lead to effects in what we call the periphery, that is out into the body and the nervous system, and they lead to weakness, coordination issues, and paralysis ultimately. So where do neurons degenerate? Well, the first thing I want to just remind everybody, or teach everybody if you've never learned it, is that the nervous system is broken up into 2 major divisions. The central nervous system, composed of the brain and spinal cord, shown in pink here in the center, and then the peripheral nervous system that is shown in the yellow lines, which would be the nerves that feed information out to muscles and to organs, but also bring sensory information in, and that's just as important to the body's appropriate functioning. When we look at most neurodegenerative diseases, we find that it's the central nervous system where they manifest the most. And so most neurodegenerative diseases are CNS focused. That is, we have aspects of the central nervous system, brain or spinal cord, that are being significantly impacted, especially in the brain. So disorders like Alzheimer's, the other dementias, Parkinson's, multiple sclerosis, and Huntington's all start in the central nervous system, as does ALS. But when we look into the peripheral nervous system, there are less disorders, but they typically are also just as damaging to the ability to control the body and eventually leading to paralysis. That would be the spinal muscular atrophy, the Guillain-Barré syndrome, and ALS as well. So you might wonder, why is ALS up there twice? ALS first manifests within the brain and then spreads progressively out into the peripheral nervous system nerves. When we look at typical symptoms of these neurodegenerative diseases, it's based on what particular regions of the nervous system are impacted. And looking at the CNS degeneration, that is aspects of the brain, there are many different proposed mechanisms for this degeneration or loss of function. And so here we have abbreviations for like Alzheimer's disease, AD, Parkinson's disease, PD, Huntington's disease, HD, ALS, frontotemporal dementia, and so forth. And what I want you to notice is that to some extent, or varying extents across these disorders, they have overlapping mechanistic problems that arise. Things like DNA damage that's not appropriately repaired, cells that become more silent and not active, we call that senescence, proteins that build up, other aspects of the genetic information that are not appropriately being cared for or repaired, and even the cellular powerhouses of our individual cells called the mitochondria that produce the primary chemical energy source called ATP can also be affected. So the underlying reasons for that are not absolutely clear, but again, we think that it's normal aspects of aging, but just accelerated to varying extents across these disorders is one possible hypothesis. But looking at CNS degeneration specifically, we see that memory is massively affected. The difficulty forming or accessing existing memories could be a major issue. Emotional state, that is, mood and behavioral shifts can occur, as we saw with the initial patient who suffered from Alzheimer's, initial discovered patient. And then declining sensory perception, so you have less ability to bring in the world around you through sensation. Loss of identity is one of the most debilitating of the various symptoms that one might experience. And then tremors in some disorders. Parkinson's is known, of course, for the tremors that it causes, but even severe forms of Alzheimer's disease will lead to tremors. When we look at peripheral nervous system degeneration, it's also highly impactful. It can affect sensation and cause phantom sensations like tingling in the extremities. It can cause declining leg strength, and that, of course, will affect walking and gait. Increased falls come from that. And then progressive decline of muscle movement, and then that eventually leads to muscles that slowly decay away, that is atrophy, and eventually the loss of control of the body, which we of course call paralysis. When we look at the most understood of these disorders, Alzheimer's, there are two major pathological hallmarks that are often pointed to as a potential cause, and I'm saying potential for a reason that Professor Fabian Fine will describe later, and those are things that are different about the brain tissue in the patients who are suffering from Alzheimer's disease, and that is increased levels of protein buildup, specific proteins that are already there, normally functioning, but then they become either non-functional or produced at too high a level. The first of which are amyloid plaques, and they're shown in this diagram as these large balls outside of the neurons. This area would be the neuron's somata, or the nucleus is inside, and then these long branch-like structures are the processes that communicate information into the neurons or communicate out to other neurons in most cases. But we start to see a proteinaceous buildup outside of the neurons that normally would not be there in healthy brain tissue, and we refer to those as amyloid plaques. This amyloid plaque is built up of a particular protein, amyloid beta peptides, that are normal functioning proteins associated with communication, but they are not regulated appropriately in Alzheimer's patients. When we look at a photograph of an actual brain tissue, these structures you see here are the amyloid plaques, and the little blue circles would be the nuclei or the nucleus of the individual neurons that are in that area. Looking to the right, we see the other hallmark of this, and that is the neurofibrillary tangles. That is, extra proteins that are building up in an irregular fashion inside the neurons, And these are intracellular aggregates of a particular protein called a tau protein that helps to support normal cell structure and keep the neuron holding its complex shape. If you had those proteins disappear, a neuron would not be able to make all those branches and be able to communicate. It would become a spherical round ball, essentially. When we see a more realistic depiction of what these neurofibrillary tangles look like, and the name neuro, of course, is neuron. Fibrillary means fibers that are tangled up and twisted up. And when we see them in actual brain tissue, this is human brain tissue, graciously shared by Dr. John DeWitt over at the VM Medical Center. These are like nuclei of individual cells, but you see that we have these neurofibrillary tangles that have coalesced and started to twist up on themselves. That's what we refer to as the tangles. The other tissue around, while it has degenerated, doesn't show those tangles. So it's not everywhere evenly. It's certain neurons that are impacted more than others, potentially. So the pathological hallmarks of Alzheimer's disease are many different malregulated processes in the buildup of proteins, and ultimately it leads to lots of different major impacts. The immune system detects that there is something wrong and starts to attack potentially living cells as part of a process of inflammation. Inflammation is something that we need to recover and repair our bodies, but in many cases it can get malregulated with, say, autoimmune disorders like multiple sclerosis, but also in disorders like this, we have too much inflammation occurring. That can, of course, lead to cellular stress when it's being attacked by its own immune system. Blood vessel damage as well, the surrounding blood vessels, so that means that blood will not flow in appropriate fashion. And the mitochondria, the powerhouse of the cells that produces ATP, will degenerate and not function appropriately, and the cells are starved for energy. That eventually leads to cell death and eventually loss of brain tissue, replaced in most cases by scar tissue. When we look at current treatments for neurodegenerative diseases, not just Alzheimer's, but other neurodegenerative diseases, as I mentioned earlier, because these occur at a later stage, when you're fully diagnosed, you've had potentially decades of buildup of these issues. It's very difficult to treat. I just want to be honest about that up front. But scientists are working towards earlier cues, earlier means to diagnose, say, blood markers and so forth that are less invasive, and also imaging techniques to see changes in the brain. So early diagnosis is key. If you have any concerns, you should talk to your clinician to get help there. Once somebody has maybe a diagnosis, they should work with that clinician to manage any neurotransmitter levels. In many cases, neurotransmitters may get released too much or too little, and they also can then manage the symptoms. If you're having tingling, something to reduce that kind of phantom sensation potentially. Other things that can help to prevent the worsening or to slow, at best, the worsening of the disorders are reducing stress. And that means physical stresses, that means chemical stresses, that means emotional stresses. Easier said than done. Reducing inflammation. So in some cases, the treatments will include reducing immune system response. Improved sleep helps to reduce stress generally and helps metabolic processes as well, as does an improved diet. And staying mentally active is key, as Professor Fabian Fine will describe later. And then finally, remaining physically active helps to reduce the stresses that our body experiences as well. So with that, I would like Professor Fabian Fine to continue our discussions.
So there's the prevalence of neurodegenerative diseases and also their irreversibility that make them such traumatic events in families, particularly, as Adam mentioned, caregivers who have to take care of people whose mental state declines. This is why mental or neurodegenerative diseases are topic to numerous research investigations, including our group. So I would like to give you a little bit of a scientific background with regard to neurons before we go into our research, or our students go into our research project. First, some good news. Memory loss doesn't necessarily mean that you have a neurodegenerative disease. There's just a natural plasticity to our brain, and this is basically the saying, use it or lose it. So if you learn a second language and you never use it, you will lose it. You will forget those words. And this is this natural plasticity that we have in our neurons. So neurons in our cortical areas form these spines, and these spines, when they're activated, or neurons when they're activated, they form these spines, and that forms your memory. As long as you use this memory and you activate your brain regularly, these spines will strengthen, and you will have regular access to this memory, your words, if you learn a second language. If you don't apply this knowledge, if you don't think about these pathways for a long time period, then they disappear again, and this is the plasticity of the brain. So many people, when they retire, for example, I hear that all the time, they say, oh, I'm getting dementia because I'm losing my memory. It is not that they're losing their memory, it's just that their lifestyle changes. So they're exposed to different things and no longer to what they were exposed to during their work life, so they tend to forget what they've worked with. But then therefore learn other things. So to keep our brain active and to think about things, do crossword puzzles, educate yourself, is certainly a good way to maintain brain health. And forgetfulness is not necessarily neurodegeneration. Although if there is neurodegeneration, forgetfulness is one of the first signs, as the structure that is responsible for learning and memory. The hippocampus is one of the first structures to go. So let's talk a little bit about those neurons in both the central and peripheral nervous system that so irreversibly degenerate during disease process. So first of all, I would like to introduce you to the structure, basic structure of a neuron, because that will be important for you to understand when we talk about the research. Neurons have a soma in which the nucleus is located, and then depending on what kind of neuron it is, they may have dendrites here coming from the soma, where we have axons from other neurons that send input into this dendrite and then send it down the axon. These are the projections to the synaptic terminals here. And now these synapses will communicate either to other neurons or to muscle cells, for example, if it's at your neuromuscular junction, and send this information to those effector cells that then respond to this. Axons don't stand alone. In order to conduct signals down the axon, they need insulation, and you can see this here. And this is, for example, in this case here, Schwann cells that insulate those axons and allow these signals to travel down. And we call these Schwann cells are very large glia cells, which is why we call them, they belong to the macroglia. There are also other glia cells. They are more the glia cells of the immune system. You will learn about those as well. And these are called microglia. This is usually here in the vertebrate or mammalian system. In invertebrate systems, these glial cells look a little bit different, but they have just a similar effect. They insulate the neuron, and you will learn shortly that they also have a very important other function. So this neuron that looks a little bit like a burning neuron is surrounded by these glial cells, and you can see that they are folded onto each other. And these folds when they unfold, allow this glial cell to have a bigger volume, as you will see shortly. So, as you can see, these glial cells are very, very tightly connected with the neurons, and if we lose those glial cells, it will damage the cell membrane of the neuron, and the neuron loses its functionality. And you can imagine this when we look at this metaphor here that we have our electric wire. Our electric wire needs to be insulated, and we have these copper wires in there that conduct the electricity down the wire. If we were to take this insulation off of our wire, our electricity would just leak out of it, just like the water would leak out of our garden hose if we have holes in our garden hose. So this would be very detrimental for neurons. And as you can see in this example here, this is an invertebrate glial cell that surrounds a neuron here. That in the center is the neuron, that's the mitochondrion. And you can see that these glial cells are held together. And you can imagine this like Velcro. Velcro that holds these arms of these glial cells, by the way, that's one glial cell here, together, and forms this very tight connection between the neuron and the glial cell. And you don't see this in these pictures, but there are many receptors and many cellular processes that, with regard to signaling, that go on, and they can only go on if this system has integrity, if this system is intact. If it comes apart, these signaling molecules and signaling cascades will be interrupted. Here you can see a mammalian, that's an optic nerve, and what you see in the center of this optic nerve, so that's the nerve here, this is the glial cell that surrounds this nerve, and you can see the tight insulation. And what you see here, these little round organelles, these are microtubules. And microtubules, what you will learn is, seem to play a very important role in neurodegeneration. And we are about to understand what happens in neurodegeneration. So microtubules seem to be affected by this. And you can imagine the microtubules look just like these wires these copper wires and this electric wire here. They are long filamentous structures, and we have many of them, and they have many functions within the neuron. I would like to, in order to explain the function of the neuron and how a neuron conducts signals, I would like to take the metaphor of the electric lamp. So everybody is familiar with an electric lamp. So you have your cable that you plug into your outlet, and then it goes up to your individual lights here. If you put power on it, you have your on and off switch. If you put power on, then your lights turn on. You also need your screws and everything in order to hold your light together. If you don't have those screws, your light won't work because nothing is attached. The cables are not attached. These holders for the light bulbs are not attached. Same is true with neurons. So we have our soma here. We have our axons that make synaptic contact onto our dendrites. That is our power plug. Our power is then in our neuron and is conducted along the axon. This is our power cord along the axon. Now we come to our synaptic terminals, and here we then have action going on. In this case here we have postsynaptic dendrites, and these will be activated. Here we have our lights that are turned on. The screws in our glial cells here are these desmosomes. And I would like for you to take a closer look on these desmosomes. And this is actually something that we have just submitted that has not been described previously. We didn't know that before we investigated this. What you see is that the glue, this Velcro-like connection here, these are desmosomes, are regularly attached to these microtubules that you've just seen in this optic nerve. Previously, it was always believed that they adhere to intermediate filaments, which is another filament of the cytoskeleton of the neurons, but they are not. They are clearly aligned with microtubules. And you will understand the significance when we show you our research. Microtubules are critically important players, not just in neurons, but in all the cells of the body. They are basically the skeleton of our cells. They have many roles. One of them is a skeleton, like this skeleton here in the hand of Marie Curie. If this is our neuron, you can see this beautiful drawing here of those microtubules within the axon. Here's a growth cone, as does axon. As axons grow out, these microtubules help the axons grow out. Then here you can see the dendrites. Here, again, they provide stability for those axonal or for these dendritic processes, but also for the axonal processes. Imagine in this picture here, the microtubules are no longer there. This neuron collapses. This neuron collapses and loses its connections to the other neurons and also to the postsynaptic and presynaptic structures. So, with other words, they are, if we were to magnify this out and we look into this neuron, these microtubules here indicated in green are very important players for the structural integrity of the neuron, but also for transport along the neuron. So neurons are metabolically very active and they produce a lot of proteins. So they have to transport them from here, where the proteins are produced, all the way to the axon. And this distance can be a meter long in the motor neurons. And in blue whales, they are very long. So with other words, the microtubules are the highways or transport roads within the neurons on which they rely in order to keep those neurons functional. Microtubules have a dynamic structure, and they have to have a dynamic structure because sometimes, as I've shown you in learning and memory, you have to form a structure and then you have to retract it, and you have to form another structure and you have to retract it. And they can do this because they're dynamic structures. They can assemble and disassemble, and these are these little building blocks here. And you can imagine this very much like your Lego blocks. You know, you can take some off at the bottom and you can put some on at the top, and then you can have some rubber band so to prevent it from falling apart. And neurons have that as well. And what stabilizes neurons is tau protein, which you've probably heard about in context with Alzheimer's disease. So in mature neurons, these microtubules don't disassemble, but they have capping proteins that keep them stable because this assembly and disassembly costs energy for the cell. And so when you have mature neurons, they will be stabilized with these capping proteins. And again, here you can see the microtubules. I want for you to recognize them as you will see them in our research pictures and what happens to those microtubules during neurodegeneration. I also would like to introduce some chemicals that interfere with the integrity of microtubules, and one of them is ethanol, or the ethyl alcohol that we drink that's in our alcoholic beverages. And these experiments here have been made in zebrafish. And when I first saw them, I was really amazed by these findings. Zebrafish, as they develop, they rely on their microtubules on pulling the embryo over their yolk sac. And this is what you see here. These are the microtubules. I didn't bring the images of the cells of the yolk sac because I would like for you to focus on those microtubules. You see this beautifully filamentous aligned yellow or green stained lines, which are all microtubules. If a zebrafish has been exposed to ethanol in their medium, and it was 1% ethanol, you can see that those microtubules disintegrate. And you can only see pieces of microtubules, but they are no longer filamentous structures. And if I put this right next to each other, you can see the difference. The microtubules in control preparations and the microtubules in alcohol-exposed organisms, which leads in the zebrafish to abnormal development that are, depending on which stage of development they are exposed to this ethanol, is often not compatible with life. And we have a similar syndrome in humans, and I would like to mention that microtubules are highly conserved throughout the phylogenetic tree. So single-celled organisms have the same structured microtubules than humans have. They are very, very highly conserved. And if you now look at the human brain that at the infant stage is exposed to alcohol in utero and of the mother. Then you can see here, this is an infant. These are infants of comparable ages that have died of, in this case here, fetal alcohol syndrome, and in this case here of unrelated alcohol-unrelated conditions. You can see that the brain is highly impaired due to this exposure to alcohol. Similar findings were made in the mammalian brain of rodents, where you can, in this cortical area here of the control animal, you can see that there are hardly any degenerating neurons. The degenerating neurons are shown in black here. They are black stained. Whereas mouse that was exposed to ethanol in its drinking water has a huge amount of neurons that start to degenerate in the cortical area of these mice. Similar findings were made in neurons of the hippocampus. Again, this is this area that is important for learning and memory, that neurons that normally have a lot of neurites that they form and make a lot of synaptic contacts now no longer have the high number of dendrites, but these numbers are reduced. Now, if we consider that there are microtubules within those neurites, we hypothesize that it is the alcohol that leads to the dismantling of those microtubules and may explain why we have alcohol-induced dementia in patients who drink a lot of alcohol. So, I've mentioned this tau protein previously, and as Adam has also mentioned, we have these fibrillar tangles in the mammalian system that we see in the brain, and these consist out of this tau protein. But what is tau protein? Tau protein is a protein on microtubules. It's a so-called MAP, a microtubule-associated protein that stabilizes microtubules. And the current hypothesis is that these tau proteins phosphorylate, and we have these hyperphosphorylated tau proteins that then tangle up. The microtubule therefore loses its stability and then disassembles. However, this hypothesis has been there for 30 years, and research has not made significant progress for developing any cures based on this hypothesis. So the question is now, is the formation of tau clusters the cause for Alzheimer's disease, or is it a result out of the disassembly of microtubules? And so we have decided to investigate this a little bit more in an invertebrate model organism, and we'll show you our findings. Invertebrate model organisms are very important because if you look, for example, here at the brain of a human, and here of the brain of our model system, which you will learn shortly about, you can see that the tissue preservation in the human model system is just much not as good as it is in the invertebrate system. This is a degenerating brain, and we can still see the neurons quite clearly and the cellular perimeters of those neurons. So it's also the fact that these neurons are much bigger, and we have a way more simple brain in invertebrates that will allow us to pinpoint the earliest stages of degeneration. When does degeneration first occur, and what do we see in these early stages? That allows us to pinpoint down those processes that initiate neurodegeneration in our invertebrate system. And what our students will now introduce to you are things that have not previously been observed, and we've just submitted this publication. And I would like to give the microphone now to Abigail Delpy, who is a senior in neuroscience and has been participating in this research for 2 years now. And here you go.
Thank you. So, our vertebrate model organism for this research is the Central American wandering spider. Now, these spiders, they're not your average creepy crawlies that you find in your house. They're about 11 centimeters in leg span, so that's about the size from the base of my palm to the tip of my longest finger. That's pretty big. Before I started doing research, I was kind of scared of them. And like your average spider, they have two main body parts to them. The prosoma, which has the head and the legs, is the front end of the spider. And the back end is called the opistosoma. Now, we concern ourselves mostly with the prosoma since it houses the spider's brain. And yes, I said spider brain. They do have brains. Shocked me too. And they look a little bit like this. Now, the advantage to using these spiders' brains in our research is that they have larger neurons, which are 3 to 4 times larger than a mammalian neuron. And these neurons are grouped into ganglia that are easily identifiable, as there are few of them. And you can see here, these are a representation of the ganglia in the spider. Now, 8 of them are each attributed to one of the spider's legs, and they are labeled here, and their coherent pairs are on the other side. These ganglia send their axons into the legs to help the spider walk and hunt, much like we have nerves that go into our arms and legs to help us function on a daily basis. Now, I'll pass the mic to my colleague, Taylor, who will talk a little bit about their behavioral processes. She's a junior neuroscience major.
Thank you. So the reason we started this neurodegenerative study on spiders was that we had noticed an interesting behavioral difference. The young spiders who sit on a vertical surface with their opistosoma upright and their prosoma downright, almost at a 180-degree angle, just like the spiders you see at home, we noticed that as they aged, a subpopulation started to droop. Their opistosoma started to droop with gravitational forces, as you can see here. They were no longer able to hold themselves upright against the vertical surface, and they would fall downward. It also progressed to a point where we could take our forceps and hold them, and they wouldn't run away. This indicated to us that we were seeing the spider equivalent to the human form of ALS, with the spiders losing all their peripheral motor function. We also noticed a difference amongst siblings. Not all siblings showed any degree of neurodegeneration. So some showed severe form of drooping and some didn't have drooping at all. In order to investigate the cellular underlying cause for the spider's inability to use their legs, we dissected the brains of young spiders and old spiders. And our first striking observation was that the younger spiders have this beautiful ivory-colored consistency, and the older spiders have this brown tint. And these were not stained in any way at all. This is their natural color. And I want to compare that to the photo over here that Dr. Weaver showed you earlier of a normal brain here, and then the brain of Alzheimer's patient right here. And we also see the same brown tint. And another finding was, or as you can see, it's noticeably smaller than the normal brain. And we also see this in our spider right here, with the older spider having a smaller brain as compared to the younger spider. My colleague, Jason Boucher, senior biology, he's been working on this project for 3 years with both Dr. Weaver and Dr. Fabian Fine. He will discuss neurodegeneration on a cellular level.
Thanks, Taylor. So what I'd like to draw your attention to first is this schematic up in the top right. As we see the right hemisphere and left hemisphere, we see each leg ganglia. And what I will tell you is each leg ganglia controls a specific leg. So if I say, here we are in LG1 in the right hemisphere, what that means is that lobe right there, that ganglia, is controlling that first walking leg on the right side of the spider. Now what I'd like to bring your attention to here, these are semi-thin sections, and these are 1/1,000,000th of a meter thick. And what we've done is we've stained them And this stain, what it does is it shows in purplish-dark blue proteins. Now I'd like to go into a little bit of what we see in the semi-thin section. So as we see these beautiful neurons, and these neurons have well-defined kind of perimeters, and we also have the neuropil here. And the neuropil is where the axons from the neurons come out to form nerves which send action potentials down to innervate the actual leg. Now, this part right here, you may say, oh, well, why isn't this stained? This is actually very common. It's also common in humans. This is related to the ventricle, or this is the ventricle, the equivalent of the ventricle in our brains ourselves. So now, don't all gasp at once when I show you the old brain. Right? Crazy. But what I want to bring your attention to is the difference in color. As we can see, and really the difference in color in the neurons and in the peripheral of the brain. First, the neurons seem to be very lightly colored, which we kind of wonder, well, where'd all the protein go, right? Which I will get to later. But the next thing we see is this other purple color kind of in the neuropil and also where it filled in with the neurons. And this is the scar tissue. Now, the scar tissue we hypothesize to be lipids, but this is degenerated neurons and axons that have been, and we hypothesize to be lipids that are left behind. So the question I posed just a second ago, where did the protein go? Well, what we see here is on the outside of the brain, we have these macrophages. And in the outside of the old brain, we have copious amount of macrophages, and they're much darker in color, which we can show has the protein from the interior of the brain inside of it. Now what I'd like to bring your attention to is we counted the area of the spider brain, and what we found was a significant difference in the amount or percentage of scar tissue. In the young brain, we had an 8%, the area of the young brain that had scar tissue in it was 8%, compared to the area of the old brain that had scar tissue in it was 38%, which is a 30% increase, which is incredibly significant. Next, I'd like to kind of just highlight the healthy neurons versus the non-healthy neurons. As we see, these are a characteristic golf ball or tennis ball-like structures, very plump and colored great. Here, we see a very lightly stained neuron and also a large amount of scar tissue here as well. Finally, I'd like to point out the neurolamella, which is here. In the young animal, it's very straight and looks healthy. Compared to the old animal, it's curved, which my teammate, Josh Weiss, who's been working on this project for 3 years, will talk about.
All right, so as Jason has pointed out, there's significant neurodegeneration, scarring, and a disintegration of these membranes or neurolamella in these aging spider brains. And this encouraged us to look into this neurolamella a little bit more in detail. So to reiterate, in the 1-year-old spiders, which are a bit healthier and functioning normally, we can see that this neurolamella, or this membrane, is very well intact and it's straight. When we skip ahead to the 3-year-old spiders, we can now see that this neurolamella and these membranes are overwhelmed with this darkly stained cellular debris. And this was significant because now we wanted to find out, well, we know that this cellular debris is being transported across this membrane. As we can see here, when you zoom in on this neurolamella, what we find are these very small microchannels where the cellular debris passes through the neurolamella and is then picked up by these macrophages with the cellular debris now stained in yellow instead of that dark pink. And this led us to our next important question, which is, well, where is the cellular debris originating from? We know that it's accumulating, it's passing through the neurolamella, and it's being picked up by these macrophages. But again, where is it actually coming from in the tissue? So when we look back at these 1-year-old healthy spiders, what we can actually see here are these healthy cells, these circles, which are then surrounded by this macroglia, which appear to just be the outline of the cells, but they're actually separate cells that support the neurons. And we can see here that even in these 1-year-old cells, these macroglia, these crescent-shaped cells, are starting to accumulate this cellular debris. So to bring it all together, what we've now found out is we've outlined a mechanism of neurodegeneration even in these younger spiders, which has previously not yet been described. To talk about this in a little bit more detail, I'm now going to introduce Anna Akins, a junior neuroscience major who's been working on this project for over a year and a half.
Thank you, Josh. So interestingly, the immune system of our invertebrate organism is very similar to that of a mammal. This includes that when there's an increase in neurodegeneration and inflammation, there's also an upregulation in the immune cells. And these immune cells are needed because they help to repair the damage that was done to the brain by the inflammation and neurodegeneration. And the immune cells which I'll be discussing today are macroglia, microglia, granulocytes, macrophages, and multinucleated cells. Now, the macroglia are not your average cell. As you can see here, this is the nucleus of the macroglia. It is the dark purple triangular shape. And as you can see, it is not surrounded by the typical circle structure of a cell, but rather the macroglia then extends out into the brain. I like to think of the macroglia similar to a tree. The nucleus is the base or trunk of the tree that is grounding the cell, and its extensions are the branches. And those branches then go out and surround and insulate each individual neuron. The microglia are located in both the invertebrate and mammalian systems, and their job is to collect all the debris and transport it out of the brain. The granulocytes are these dark purple granular-looking cells, and they communicate with these other immune cells and share enzymes with them that help to break down that debris. The macrophage, which is this large cell right here, is very similar to the microglia in that it also collects all that debris and transports it out of the cell. However, when it reaches the outside of the brain, there we go. When it reaches the outside of the brain, it then fuses with the other macrophages and creates these multinucleated cells right here. And we will discuss those multinucleated cells in more depth later on this evening. We decided that the next step was for us to look at the spider brains under the electron microscope so that we could see them in more detail. So when we did this, we realized that the macroglia actually fold in on themselves and create these finger-like projections located here, here, and here. But these finger-like projections of the macroglia are connected to each other via cell adhesion junctions known as desmosomes. There are multiple types of cell adhesion junctions. However, the desmosomes are the strongest ones. We also noticed that there were microtubules located in these finger-like projections when they were near the desmosomes. Once again, here is your finger-like projections, your desmosome right here, and these smaller circular structures are your microtubules. So we have microtubules here, here, and here. Now these microtubules and desmosomes act like a zipper. When there is no neurodegeneration present, they're all zipped up and ensure that the macroglia remains intact. However, if they were to come undone due to neurodegeneration, the macroglia would eventually unravel, and this would severely damage the cell membrane of the neuron, because the macroglia essentially sits on top of the cell membrane. The microtubules and desmosomes are also comparable to a pleated skirt. Now, what holds together the individual pieces of cloth of this skirt are the stitches. And our stitches are these desmosomes and microtubules because they hold together the individual finger-like structures of the macroglia. If the stitches were to come undone in the pleated skirt, this would cause the skirt to fall apart. Likewise, if these microtubules were to disassociate, this would lead to the desmosomes disassembling, and ultimately the macroglia would unravel. And now my colleague Sean Clem, who is a senior neuroscience major and has been conducting this research for approximately a year and a half, will discuss our findings on the unraveling of macroglia.
Thank you, Anna. So as Anna has just described, these desmosomes function much like the glue for our nervous system. So as you can see here, we have a centrally located desmosome and these two associated cells next to it. And they're also super important for keeping the integrity of the nervous system and the shape of the nervous system, as well as the shape of the neurons. So the question you may be asking yourself is, what happens when these desmosomes come apart? And this is exactly what we see in the first early stages of neurodegeneration within our specimen. So as you can see here, this is an early stage of neurodegeneration within our spider brains. So there's several things going on here. So first, as you can see, this is the macroglia that has been described previously. And this is also part of the macroglia. However, it doesn't look organized anymore. It's forming into this classic zigzag structure that we come to see. And also, the circular shape of the cell is not maintained. It fills into these jagged intrusions and protrusions from the zigzag structure of this macroglia. So I'd like to turn your attention to a healthy set of neurons. So we can see here that the macroglia follows the perimeter of the cell, leading to this golf ball-like cell that we get to see here. But when we turn to the degenerating neurons, we don't see this. Again, we see this zigzag structure forming, leading to the malformation or the difference in shape that the neuron, as we can see here, it's not golf ball shaped or round anymore. But we also see that the volume of the macroglia is increasing as it disassembles. And again, I'd like to reiterate the example of the pleated skirt. So if we were to remove one of these stitches, the integrity of the pleated skirt isn't there anymore. Much like when we disassemble these, or when these desmosomes disassociate, we get to see the zigzag structure, which is the first sign of neurodegeneration. Now again, we see the healthy cell here surrounded by its macroglia, and then when we look at the degenerating neuron, we see the thickening of the macroglia also leading to the increased volume of the macroglia, spearheaded by the disassembly of the desmosomes. But we also see that the cytoplasmic structures within these neurons is becoming depleted. Now, as Dr. Fabian Fine has presented earlier on in this presentation, microtubules aren't just important in keeping these desmosomes together. They're also super important in cell transport. So another question that you may be asking is, what happens to these microtubules that disassemble in cell transport? And again, we turn to our degenerating specimens. So I know that there's a lot going on here, but I'm going to orient you quickly. So this is our degenerating neuron, as we can see here. We can see scar tissue forming above and below this neuron. We can see that the volume of the macroglia is actually increasing, and you can see that happening here. You can see the axon of this neuron degenerating along with its associated microtubules. And then there's something else that you can see. These are actually transport vesicles, and they normally transport down the axon, and you can see that in the schematic. They can normally transport down this axon and get to where they need to go. However, they're stuck at this region of the neuron known as the axon hillock, and that is right about here. So the analogy that I would like to have you think of is the analogy of a train station. So think of the people on the platform waiting for their train. They get on their train and they go down the train tracks to where they need to go. So in this example, our transport vesicles are the people standing on the platform waiting for their train. And the train tracks are the microtubules on which they travel. Now, when these microtubules dissociate, this is analogous to our train tracks breaking up. So these people, or the vesicles, can't get where they need to go. And similarly, you would have people stuck on trains downstream from the train station. And we can see that here too. So these are transport vesicles that are stuck within this axon due to the fact that the microtubules are degenerating. And yet again, I'll bring one more analogy. So the analogy that I like to think of for degenerating microtubules is the rubber band analogy. So you take a rubber band and cut it in half. You hold each end of the rubber band in each one of your hands and give it some tension. This is what a normal microtubule, or this is what you can think of that a normal microtubule would look like. It's straight, long, and stable. Now start to decrease the distance of your hand when holding that rubber band, or even release one of the ends of the rubber band. And this is comparable to what you see in our degenerating nervous systems. You can see that this wavy formation, which is where microtubules would be, is happening. Now it's important to state that at this stage of microscopy, which is compound light microscopy, we can't see microtubules. However, we can see the transport vesicles here, and in our brains we can connect that they're on or along something. Now I would like to have my colleague Colin Radican explain inflammation of the nervous system. Colin is a biochemistry major and has been working on this project since the beginning of last semester.
Thank you, Sean. So in going back to the beginning of the presentation, we had discussed how as spiders age and the neurodegeneration occurs, typically the brain will turn to a brown coloration. But of particular interest, what we find more interesting is the dark brown accumulation of cells on the outside. So what these represent is the connection between the peripheral nervous system of the leg cells and the central nervous system of the brain. And if we look at the images on the bottom, we could typically see where the nerves transect through the neural lamina between the central nervous system and the peripheral nervous system. So in comparing a young and old cell, we typically see that in a young one, it's fairly clear, and its white coloration suggests a picture-perfect happy cell, no accumulation of anything. But in the older cell, we see that there's an accumulation of dark blue cells throughout. And then when we look at this underneath a higher-powered microscope, it is revealed that there are actually immunocells, such as glial cells and macrophages, that are attempting to clean up the neurodegeneration debris that occur from said process. This is known as an inflammatory response as this neurodegeneration occurs. So in discussing further, we get a closer view of the neurolamina as it separates the central nervous system and the ganglia, and in particular, the high concentration of macrophages that occur as they bud off from this neurolamina membrane. So in comparing young and old again, as you can see here, the young on the left and the old on the right, we see that there's a high macrophage number along with immunocells, but we can also see that they're clear, so there's nothing being done with them yet. But then later on, we actually see that there's an accumulation of debris over time as they're older. This is an attempt to preserve the longevity of the cell as this neurodegeneration occurs. And to discuss this further, I'll be passing this on to Senior Julia Ogg, who is a health science public health double major who has been working on the project for the last 2.5 years.
Thank you, Colin. Following up on what Colin was discussing, we wanted to take a closer look at what was happening to the debris as it exits the brain tissue and crosses the neurolamella. Upon further observation, we observed that these debris right outside the neurolamella are collected by the macrophage cells, as seen in this photo. This begs two questions. One, where do the macrophage cells go? And two, what happens to the debris inside of the macrophage cells? On the left here, you can see the individual macrophage cells collecting the debris right on the outside of the neurolamella. These individual macrophage cells then fuse together, creating multinucleated cells. Here's a multinucleated cell shown here. Multinucleated cells can be composed of as little as 2 individual macrophage cells fusing together to over 500 individual macrophage cells ultimately fusing together. On the right here, you can see a multinucleated cell and 2 individual macrophage cells full of debris. Those 2 individual macrophage cells are fusing with this multinucleated cell. And you can see in here individual nuclei, which indicates that those were once previously individual macrophage cells that have fused together over time. Interestingly, we can see debris clearly in the macrophage cells, as seen over here as well. However, we don't really see any debris in the multinucleated cells. This suggests that the multinucleated cells work as a waste disposal system, essentially eliminating all cellular debris waste from the environment. On this slide, I'm going to give you a little summary. So taking a step back, remember that this is what a healthy neuron looks like. Notice the round borders and the macroglial cell surrounding it. Also notice the healthy neuron and the pigment inside of it. Now, in this image here, you can see one of the first signs of neurodegeneration. That is the unraveling of the macroglia, as seen here. The unraveling of the macroglia leads to it helps contribute to the progression of the emptying of neuronal contents inside a neuron, which in turn creates the macroglia that surrounds the neuron to swell. That is shown in this image here. Notice that this is the macroglia, and it's very thick and swollen compared to the healthy brain tissue. Also notice that the neuron inside of the neuron is a lot less pigmented than in the healthy brain tissue. I also want you all to understand that the neuronal contents in here that are being drained from inside the neuron are the debris that I was just discussing. In this image here, the neuronal content, now debris, is stained yellow. You can see it exit the brain, cross the neural lamella, and the macrophage cells pick up the debris on the outside, and then the macrophage cells then fuse into the multinucleated cells, which eliminate the neuronal content, now debris, from the environment. So this whole process begs the question, what happens to the empty space, or the space that's now empty, that once occupied the neuronal contents inside of the neuron? In this image here, you can see that this was once a previously healthy neuron, and it is now being gradually filled with a pink granular-like substance. This is the formation of scarred tissue. We are currently looking into the formation of scarred tissue as a research group, but we have reason to believe that those pink granular fatty substances that are filling the area are fatty lipid material. But again, we're still looking into it. Now I would like to invite Dr. Fabian Fine back to the stage.
So to summarize what we've just described to you, here's our schematic drawing where you can see the progressive neurodegeneration as we see it in invertebrate organisms from the earliest stages. We have our intact neurons, neuron 1, neuron 2, with the Golgi apparatus, microtubules that transport proteins down and up the axons. We have our macrophages on the outside, or our macroglia, here around the neurons, and our macrophages on the outside that normally just transport normal debris out of the nervous tissue. Something goes wrong in the degenerating brain, and it is those junctions here that go first. It is those microtubules, those desmosomes that go first and lead to the unraveling of the neurons. And it is this fact that causes me, honestly, sleepless nights, because now we have a process where we can focus on and see what is going wrong during these processes, what leads to the unraveling of those glial cells, and can we prevent this from happening in early stages of neurodegeneration. So we are currently teaming up with UVM Medical Center, who have human brain tissue available for us to fix and investigate in collaboration with them, and see if we can see similar cases of degeneration there. One thing that I want to bring your attention to is that as this neuron degenerates, because it shares the boundary with this neuron, it also causes the degeneration and activation of these macroglia in this neuron, and this is known as the so-called penumbra effect, and that makes the neurodegeneration progress faster and faster and faster throughout the brain and leads to this degeneration. And there is a mammalian equivalent to this, and it's called the glymphatic system. It is a disputed system, but our system supports this. The difference in the mammalian system is that we have those astroglia, these are our macroglia cells, these are our neurons. They are believed to let cerebrospinal fluid into the interstitial space between neurons in our brain and wash our debris. Most prominent, there's a beta plaque out of our brain, and interestingly, this only happens at night. This supports the hypothesis that sleep is critically important for the clearing of debris out of the brain, and if we suffer from sleeplessness, that this might actually promote neurodegenerative processes. So to summarize our findings here, neurodegenerative diseases are on the rise, they're irreversible, pointing out how important it is to find cures and understand the mechanism. Change in memory is not always a sign of neurodegeneration. There is evidence that alcohol might actually induce neurodegeneration through the disassembly of microtubules. Invertebrate model systems may hold the key to gain a better understanding and then compare what we find in invertebrates with the human system. And we have strong evidence that sleep is important for the health of our brain. We would like to thank the Vermont Biomedical Research Network for supporting our research. This research was conducted by the neuroscience research team and classes, and we've just submitted this manuscript. We would like to thank St. Michael's College, VPAA, Jeff Trumbauer, for supporting this research and the students, the St. Michael's Biology Department, Dr. Lapkowitz and Dr. Christina Chand for their feedback. And we would also like to acknowledge Dr. Ronald Fay, who has supported the students, and Dr. David and Ann McLaughlin, who have supported our lab and the biology department with informative equipment. Thank you so much. Are there any questions that we can answer?
So the question is for remote viewers, how does CTE or chronic traumatic encephalopathy relate to these other neurodegenerative disorders? And often, not always, but CTE is often associated with trauma, repeated trauma to the brain. And in many cases, that trauma, that physical trauma, accelerates that degradation and leads to these processes at an accelerated fashion in younger individuals typically. So it is the same processes, but usually earlier onset, and we see more similarities than differences. So I'm glad you noticed that. Yes.
Are you guys looking to see if, like, you're trying to find the tau protein buildup, or like look at the microtubules?
So the question again for the remote viewers is, what is the research we plan to do with human tissue? And I would say that we hope to see things with our eyes from our individual invertebrate model that others may have overlooked or assumed was just an artifact. But I'll leave it to Ruth.
Yes, that's a very good question. it is in our system for the first time that we've shown that desmosomes are associated with microtubules. At least the glial cell-derived desmosomes are associated with microtubules, which explains that if the microtubules go, why the glial cell unravel. We don't know that for the human system or mammalian systems, and this is something where we want to look at and having better fixed preparations and investigate this a little bit in more detail. Okay, thank you so much, everybody, for attending. And the next presentation will be by Alain Brizard, and he will talk about alternative energy resources in a carbon-free future. And then afterwards, on November 3rd, is Ari Kirschenbaum, who will talk about weed and the wheel and how recreational drugs affect behavioral and motor skills. Thank you so much.